Imagine, for a moment, that medicine is not merely a catalogue of symptoms, diseases, and cures, but a grand narrative of life's most intimate battleground. Deep within the human frame, in realms invisible to the naked eye, there exists a theater where triumph and tragedy play out ceaselessly. This theater is the cell, the fundamental unit of life, and, as Rudolf Virchow so presciently declared in the 19th century, the crucible of all disease. The word pathology descends from the Greek pathos, suffering, and logos, study. It is therefore the science of suffering, an endeavor to decode why bodies falter, why tissues wither, why health dissolves into disease. Once this pursuit was guided by the gross inspection of organs, the dissection of cadavers, and the recognition of structural distortions. But as the centuries turned, the spotlight narrowed from organs to tissues, from tissues to cells, and now, in our own epoch, to the infinitesimal world of molecules and genes. Where once physicians attributed maladies to humors or vague constitutional weaknesses, Virchow redirected the gaze. Disease, he proclaimed, originates in the cell. In the wake of this revelation, the microscope became not merely an instrument, but a lens into the epicenter of illness. And yet, even Virchow's audacious claim was but a stepping stone. Today we understand that it is not simply the cell as a visible unit that dictates health or disease, but the choreography of molecules within it, genes, proteins, metabolites that govern survival, proliferation, and death. Thus, the modern discipline of pathology has become, at its core, an odyssey into the intimate mechanics of the cell. It is here, in this infinitesimal cosmos, that the threads of fate are spun. A gene mutation in a stem cell may set forth the long road to cancer, a misfolded protein may trigger neurodegeneration, a deficit of an enzyme may curse a child with metabolic catastrophe. The physician who peers through the lens of pathology is therefore not merely diagnosing an ailment, but unraveling the logic of life itself as it oscillates between harmony and chaos. To embark on such an odyssey, one must first stand upon the firm ground of what is normal. For how can one recognize aberration without first beholding order? The human cell, though but a microscopic droplet of cytoplasm and organelles bound within a delicate membrane, is no simple structure. It is rather a metropolis, plasma membrane as its fortress wall, nucleus as its archive of laws, mitochondria as its power plants, endoplasmic reticulum and Golgi apparatus as its factories and couriers. Within this bustling city of molecules, signals rush like messengers, scaffolds hold like girders, enzymes act as tireless artisans, and DNA scripts the destiny of all. The purpose of this chapter, therefore, is not to drown the listener in every detail of cell biology. Such an ocean is far too vast to cross in a single voyage. Rather, it is to chart the currents most relevant to the study of disease, those principles and pathways whose disturbance yields pathology. We shall begin with the genome, the grand manuscript of life, before journeying into the mysterious world of non-coding DNA, epigenetic architecture, the quiet yet formidable regulators microRNAs and long non-coding RNAs, and the revolutionary tools of gene editing. We will traverse the housekeeping chores of the cell, the dynamics of its plasma membrane, the marvels of transport, the resilient scaffolds of the cytoskeleton, the dialogues of cell-cell junctions, and the ceaseless labor of biosynthetic organelles. We shall meet the cellular custodians, the lysosomes and proteasomes, and witness the fire of metabolism kindled in the mitochondria. And finally, we shall arrive at the higher-order symphony, where cells listen and respond to whispers of growth factors, translate signals into action, converse with the extracellular matrix, and preserve the sanctity of their lineage through proliferation, stemness, and regeneration. Each of these themes, like successive movements in a grand composition, shall remind us of a single truth, that disease is not a foreign invader alone, but often a discord within the very notes of life's music. And to heal is to restore harmony to that orchestration. With this foundation laid, let us step into the first great portal of our journey, the genome, the code that inscribes both life's marvel and its maladies. The genome. At the dawn of the 21st century, humankind achieved a feat that only decades earlier would have seemed like a dream whispered by science fiction writers: the sequencing of the human genome. Suddenly, the manuscript of our existence 3.2 billion base pairs, long was laid bare, each letter an enigmatic hieroglyph that promised answers to mysteries of health, disease, and destiny. And yet, as with all grand revelations, the unveiling of the genome did not bring closure, but rather opened a deeper labyrinth of questions. We once thought that complexity must lie in sheer numbers, that the grandeur of a human being, with its 50 trillion cells, must be reflected in a sprawling abundance of genes. But nature is a master of irony. For when the genome was deciphered, we discovered that a mere 20,000 genes encode proteins, the architects of enzymes, scaffolds, receptors, and messengers. That is just 1.5% of our entire genetic library. To place this in perspective, the humble worm, with scarcely 1,000 cells in its entire body, harbors almost the same number of protein-coding genes. The question rises like a riddle: if a worm and a human share such similar tallies, where then does humanity's vast complexity reside? The answer lies not merely in the visible text, but in the annotations, the whispers between the lines, the mysterious non-coding DNA that forms nearly 98.5% of our genome. For years, these expanses were dismissed as junk DNA, a dark matter of biology thought to clutter the script without adding to the story. But the truth revealed itself with astonishing elegance. This so-called junk is the architect of our symphony. While proteins may be the bricks and beams, the non-coding regions are the blueprint that decides how those bricks are arranged, which beams shall rise, and which structures remain dormant. Indeed, it is now understood that over 85% of the genome is transcribed, and nearly 80% is devoted to regulation rather than building. Thus, in the contrast between worm and man, it is not the abundance of building blocks that makes us distinct, but the subtlety of regulation, the timing, the context, the modulation of expression. If proteins are the instruments, then non-coding DNA is the conductor's baton, ensuring that the orchestra plays not noise, but music. And this music is composed of diverse motifs. Within the 5 major classes of non-protein coding sequences reside the very keys to genetic destiny. Promoters and enhancers, like switches and amplifiers, that determine whether a gene shall whisper faintly or roar with vigor. Binding sites for architectural proteins, which bend and loop DNA into higher-order structures, folding the genome into a dynamic 3-dimensional manuscript. Non-coding RNAs, the quiet scribes that regulate genes without ever becoming proteins themselves. Mobile genetic elements, the nomadic jumping genes that wander across the genome, leaving imprints of evolutionary history and sometimes altering gene regulation in unexpected ways. And finally, the structural sentinels, telomers and centromers that safeguard the ends and tether the middle of chromosomes, ensuring fidelity in the dance of mitosis. It is in these regions, too, that the variations of humanity are written. For despite the dazzling diversity of our species, any 2 humans are more than 99.5% identical in DNA, and astonishingly, we share 99% of our sequence with chimpanzees. The differences that shape our faces, temperaments, and vulnerabilities to disease, therefore, are compressed into less than 0.5% of the code, about 15 million base pairs, scattered like subtle brushstrokes on a nearly identical canvas. Within that delicate variation reside the most common forms of polymorphism, single nucleotide polymorphisms, SNPs, the quiet substitution of one letter for another, and copy number variations, CNVs, entire stretches of DNA repeated or deleted, ranging from thousands to millions of base pairs. SNPs, numbering more than 6 million across the human population, may occur anywhere in coding regions, in introns, or in the intergenic deserts once thought irrelevant. Most are silent, but some alter gene expression, reshaping susceptibility to disease. Still others act as neutral markers, co-inherited with pathogenic alleles and serving as genetic signposts to illness. CNVs, on the other hand, contribute to the grander architecture of diversity. By altering gene dosage, sometimes duplicating, sometimes erasing, they may sculpt phenotypic traits and confer vulnerability or resilience to disease. Between 2 individuals, such variations account for up to 24 million base pairs of difference, a testament to the vast plasticity of our genome. And yet, even with all this variation, DNA sequence alone cannot explain the divergence of human destinies. Identical twins sharing the same script may lead lives marked by different maladies. The secret lies not merely in the text, but in the editing, the epigenetic annotations that determine which genes are read and which are silenced. Here, beyond the static code, lies the living poetry of regulation, inheritance without mutation, memory without alteration of sequence. Thus, the genome is not a static archive, but a dynamic library. Its letters may be fixed, but its meaning is fluid, interpreted differently across tissues, across times, across generations. It is this interplay of sequence and regulation, mutation and epigenetics, code and context that shapes both health and disease. With this understanding of the genome's vast expanse, we step into its most enigmatic territory, the non-coding DNA, once thought to be silent, but now revealed as the stage upon which life's true complexity performs. Non-coding DNA. If the genome is a vast library, then protein-coding genes, those 20,000 slender blueprints, occupy but a tiny corner of its shelves. The rest, a seemingly endless wilderness of letters, once carried the dismissive epithet of junk. Yet, as with so many treasures concealed beneath dust, this neglected domain has emerged as the true architect of human complexity. For decades, scientists puzzled over the paradox: how could organisms as primitive as worms share almost the same number of coding genes as humans? Where, then, was the grandeur of humankind inscribed? The answer, now luminous, is that our uniqueness is not fashioned by the bricks but by the architecture, the plans that tell the bricks where to lie, when to rise, and how to interlock. In this sense, the non-coding regions of DNA are not wastelands, but blueprints and intricate choreography of regulation. Over 85% of our genome is transcribed, though most transcripts never give birth to proteins. Nearly 80% of all DNA is devoted to regulation, guiding when and where genes awaken or fall silent. Thus, while proteins erect the walls, weave the fibers, and pump the energy, it is non-coding DNA that decides the very design of the edifice. The genius of evolution is revealed here, not in multiplying instruments, but in composing symphonies from a finite set of notes. What, then, inhabits this mysterious realm? 5 great classes of non-coding sequences define its terrain. 1. Promoter and enhancer regions, those molecular stages upon which transcription factors take their positions, either igniting or suppressing the song of a gene. These elements can whisper commands to nearby genes or loop across astonishing distances to orchestrate expression from afar. 2. Architectural binding sites, where proteins latch onto organized DNA into elaborate folds and loops. In these higher-order structures, faraway sequences are brought into close embrace, creating three-dimensional theaters of interaction that no linear sequence could predict. 3. Non-coding regulatory RNAs, ghostly transcripts that do not translate into proteins but wield power as silencers, modulators, and enhancers of gene activity. Among them, microRNAs and long non-coding RNAs stand as master regulators, directing entire symphonies of expression. 4. Mobile genetic elements, the gypsies of the genome, wandering sequences that over evolutionary eons have hopped from place to place, reshaping the script as they settled. These transposons make up more than a third of our DNA, their function still partly veiled in mystery, though whispers of their role in gene regulation and chromatin organization grow louder. 5. Structural regions, the guardians of chromosomal integrity. Telomeres, the protective caps at each chromosomal end, preserve stability through replication. Centromeres, the anchor points for spindle fibers, ensure that chromosomes are faithfully parceled into daughter cells. Within these domains, vast tracts of satellite DNA repeating sequences stretching over megabases lend strength and silence, compacting chromatin into dense heterochromatin. From these sequences arises not only regulation but also variation. The polymorphisms that distinguish one human from another often dwell not in the coding genes, but in these non-coding expanses. Indeed, disease susceptibility often hinges not upon the architecture of a protein, but upon the modulation of its expression, a shift in timing, a change in abundance, a misstep in silencing. Here, too, lies the enigma of human similarity. Despite our dizzying diversity, any two people are more than 99.5% identical at the DNA level. The spectrum of individuality, disease risk, environmental resilience, even subtle differences in personality emerges from that slender sliver of less than 0.5%. Within that small fraction, polymorphisms appear in two principal forms: single nucleotide polymorphisms SNPs, a solitary letter altered, often silent, sometimes profound. Over 6 million SNPs scatter across the genome, each with its own frequency in different populations. Some alter regulation, some sit quietly as neutral markers, and some co-travel with disease-linked variants through the principle of linkage disequilibrium. Copy number variations CNVS, larger structural edits where whole stretches of DNA, thousands to millions of bases, are duplicated, deleted, or rearranged. Between two individuals, CNVs can account for differences of up to 24 million base pairs, and nearly half of them involve coding sequences, profoundly shaping phenotypic diversity. And yet, even with such variations catalogued, sequence alone cannot solve the riddle of difference. Why do identical twins, born of the same script, sometimes diverge in destiny, one falling prey to disease, the other spared? The answer lies in the subtle strokes of epigenetics, those heritable modifications of expression that require no change in sequence. Methyl groups on DNA, marks on histones, the silence of a regulatory RNA, all these edits layer atop the genetic text like marginalia in an ancient manuscript, dictating meaning without altering letters. Thus, the dark matter of the genome is not dark at all. It is alive with whispers, regulators, and architects, sculpting complexity from simplicity, individuality from identity. And within its intricacies lies the true key to understanding both health and disease. Having surveyed this enigmatic landscape, our path leads next to the guardians of expression themselves, the histones and their modifications, the proteins around which DNA coils and whose marks decide whether the text of life shall be sung or silenced. Histone organization. To imagine DNA as a straight thread is to misunderstand its essence. For if every human cell truly contained an uncoiled strand of DNA 2 meters in length, how could it be housed within a nucleus scarcely 8 microns wide? Nature, in her ingenuity, devised a solution of breathtaking elegance, the histones, tiny proteins around which DNA winds like thread upon spools. These nucleosomal structures are the first level of compaction, and yet they are far more than passive reals. They are active regulators, arbiters of accessibility, and custodians of cellular identity. The nucleosome, fundamental unit of chromatin, is formed of an octamer, two molecules each of histones HOA, HOB, H3, and H4. Around this positively charged core, 147 base pairs of DNA coil in 1.8 turns, stabilized by short linker DNA stretches that tether adjacent nucleosomes. Like pearls on a necklace, these nucleosomes thread together to form chromatin. But unlike inert pearls, histones breathe with dynamism, constantly repositioned, marked, and erased by molecular artists known as chromatin remodelers. It is in this orchestration that the dual faces of chromatin emerge. Euchromatin, delicate and dispersed, is transcriptionally active, a meadow of openness where polymerases can traverse freely. Heterochromatin, dense and silent, is transcriptionally inert, a darkened fortress in which genes slumber. To look through the microscope is to witness this contrast, light regions of euchromatin shimmering with activity, dense patches of heterochromatin locked in silence. The distinction is not static, but shifting, responding to the needs of the cell, the cues of the environment, the whispers of development. What dictates whether DNA lies open or sealed? The answer resides in a code beyond the genetic code, a histone code, etched by enzymes that modify histones with chemical marks. These marks, like cryptic annotations in the margin of a manuscript, dictate whether a gene is read or ignored. Histone methylation, lysine and arginine residues can be methylated by specific writer enzymes. The consequence depends on the site. One methylation may herald repression, another activation. Thus, methylation is not a blunt silence but a nuanced dialect. Histone acetylation, catalyzed by histone acetyltransferases HATs, this modification neutralizes positive charges, loosening DNA's grip on histones. The chromatin unfurls, transcriptional machinery gains access, and genes awaken. Conversely, histone deacetylases HDAC reverse this openness, coiling the thread back into silence. Histone phosphorylation, serine residues may be phosphorylated, with consequences as varied as activation or repression, depending on the context and the residue involved. These marks are not permanent, they are endlessly written, erased, and read by an army of proteins. Writers inscribe, erasers remove, readers interpret. Together they ensure that the chromatin landscape is dynamic, sensitive, and exquisitely tailored to the life of the cell. Alongside histone modifications, DNA methylation itself contributes to epigenetic control. Cytosines, particularly in CpG islands near promoters, may be methylated, silencing gene expression without altering sequence. This methylation is maintained through cell division, providing heritable memory of transcriptional states. Even more mysterious are the chromatin organizing factors, proteins that bend and loop the genome into higher-order three-dimensional structures. By altering spatial relationships between enhancers and promoters, they sculpt neighborhoods of gene activity, ensuring that only the right genes are activated in the right context. It is through these mechanisms that cells, sharing an identical genetic script, can play entirely different roles. A neuron, a hepatocyte, a lymphocyte arise from the same sequence of letters, yet each interprets the genome through its own chromatin landscape, through its own histone marks and DNA modifications. But as with all finely tuned systems, vulnerability lurks in complexity. Dysregulation of histone modifications and DNA methylation is a hallmark of disease. In cancer, the epigenome becomes a battlefield, tumor suppressor genes silenced by methylation, oncogenes unfurled by acetylation, chromatin remodelers mutated to chaos. Beyond malignancy, epigenetic errors are being uncovered in neurodevelopmental disorders, autoimmune diseases, and even metabolic syndromes. Unlike genetic mutations, however, these epigenetic changes bear a tantalizing feature: they are reversible. Already, therapies are in development to reawaken silenced genes or to quiet those unleashed. HDAC inhibitors, DNA methylation blockers, these represent the first steps in pharmacologically editing the epigenome, promising that disease may one day be fought not only at the level of sequence but at the level of expression. Thus, the histones, once thought mere packaging proteins, reveal themselves as guardians of genomic identity, sculptors of transcription, and sentinels of disease. They embody a truth that permeates biology: structure is not passive, but active. Architecture itself is regulation. With chromatin's hidden code unveiled, our path leads naturally to the other great players of silent regulation, the non-coding RNAs, particularly the minute yet mighty microRNAs and their more expansive cousins, the long non-coding RNAs. These molecular whisperers, though never translated, wield authority over the fate of entire gene networks. MicroRNA and long non-coding RNA. If histones and their modifications are the architects that coil and uncoil the genetic manuscript, then non-coding RNAs are the subtle voices that edit its performance after the script has already been written. These RNAs, though never destined to become proteins, wield authority over expression with a quiet yet inexorable power. They are like shadowy scribes, invisible in the final structure but decisive in shaping its meaning. Among this vast array, two classes stand out in their influence: the diminutive but potent microRNAs and the sprawling, enigmatic, long non-coding RNAs. MicroRNAs, or miRNAs, are short stretches of RNA, rarely longer than 22 nucleotides, and yet they hold dominion over entire genetic programs. Unlike the messenger RNAs that bear the code for proteins, microRNAs exist to silence, to restrain, to fine-tune. They are guardians against excess, ensuring that not every message inscribed by the genome reaches translation unchecked. The process of their genesis is intricate. Transcribed initially as long primary transcripts, mRNAs undergo a cascade of refinements. Hairpin loops are sculpted, fragments trimmed, until at last the mature single strand remains, a thread that binds to a multiprotein complex known as RISC, the RNA-induced silencing complex. Once partnered with RISC, the mRNA seeks its target among the multitude of messenger RNAs in the cytoplasm. When it finds a complement, it locks in, imperfectly at times, and suppresses translation, or, in cases of perfect match, cleaves the message entirely. In this way, a single microRNA can extinguish the voices of dozens, even hundreds, of messenger RNAs, orchestrating the fate of developmental pathways, restraining cell proliferation, or curbing apoptosis. Their influence is profound. In health, they guide embryogenesis, tissue differentiation, and immune responses. In disease, their missteps resonate with devastating consequence. A single misregulated mRNA may unleash oncogenes or silence tumor suppressors, tipping the balance toward malignancy. Others contribute to cardiovascular disease, neurodegeneration, and viral pathogenesis. Even primitive bacteria possess versions of this machinery, as though evolution recognized early that survival required not only the creation of messages but also their disciplined restraint. Yet microRNAs are only one voice in the non-coding chorus. If they are brief whispers, then long non-coding RNAs or LNC RNAs are the sweeping orations. Defined as transcripts longer than 200 nucleotides, they are remarkably diverse, numbering, by some estimates, 10 or 20 times more than protein-coding genes themselves. For years they lay concealed, buried in the vastness of the transcriptome, dismissed as transcriptional noise. But the illusion of irrelevance has crumbled. We now know that LNC RNAs are formidable regulators, weaving themselves into chromatin, recruiting transcription factors, guiding enzymatic complexes, and even scaffolding entire molecular assemblies. Consider the famous LNC RNA cyst, whose task is nothing less than to silence an entire X chromosome in females. From its position on one X, cyst envelops the chromosome in a repressive cloak, rendering it silent while its sister remains active. This is dosage compensation at the molecular level, ensuring that female cells do not produce a double burden of X-linked proteins. Here, a non-coding RNA performs an act of genetic choreography that is nothing short of monumental, the silencing of thousands of genes with a single swat. But cyst is not alone. Across the genome, LNC RNAs assume manifold roles. Some act as guides, tethering transcriptional machinery to precise loci. Others function as decoys, distracting proteins away from their intended targets. Still others sculpt chromatin by recruiting enzymes that lay down methyl or acetyl marks, thereby opening or sealing genomic regions. There are LNC RNAs that act as scaffolds, bringing multiple proteins into proximity so that complexes can assemble and exert coordinated influence. Far from being passive remnants, these transcripts are architects of regulation, manipulating transcription, translation, and chromatin architecture with a versatility unmatched even by proteins. Like microRNAs, long non-coding RNAs are implicated in both health and pathology. Their dysregulation can fuel cancer, developmental disorders, or immune dysregulation. But they also carry tantalizing therapeutic promise. To silence a misbehaving gene, one might design an LNC RNA mimic. To awaken a dormant locus, one might recruit an LNC RNA scaffold. The future of medicine may very well be written not only in proteins and chemicals, but in RNA molecules designed to harness the native language of regulation. Thus, within the grand non-coding landscape, microRNAs and long non-coding RNAs stand as the hidden governors of expression. They are reminders that the power of the genome lies not merely in its ability to encode proteins, but in its capacity to regulate the use of those proteins with exquisite precision. And it is within these whispers and orchestrations that the difference between health and disease so often emerges. With these enigmatic players revealed, we now turn to a development that transcends even natural regulation, the human capacity to deliberately edit the genome itself. From a bacterial defense system has arisen, a tool that allows us to inscribe new letters into life's script, promising cures, provoking ethical dilemmas, and opening a new era of molecular medicine. Gene editing. For centuries, medicine has been reactive, a discipline that responded to disease with remedies fashioned after the fact. But now, with the rise of gene editing, medicine has begun to tilt toward the proactive, reaching upstream into the very origins of disease, the genetic script itself. To alter DNA is to tamper with destiny, to seize the pen from nature's hand and write anew. What was once the realm of myth, the dream of reshaping life's code, has become a palpable reality, etched not with ink, but with enzymes and nucleotides. The story of gene editing begins, curiously enough, not in human laboratories but in the microbial realm. Bacteria, though primitive, have long faced the relentless threat of viruses that prey upon them. In response, evolution armed them with a defense system known as CRISPR, clustered regularly interspaced short palindromic repeats. This is, in essence, an adaptive immune memory. Fragments of viral DNA are captured and inserted into the bacterial genome, forming a library of past invaders. When a familiar virus returns, the bacterium transcribes these fragments into short guide RNAs that lead specialized nucleases, such as Cas9, to the matching viral DNA, slicing it apart with surgical precision. What nature devised for microbial survival, human ingenuity repurposed into a tool of astonishing versatility. By designing synthetic guide RNAs, scientists can now direct the Cas9 nuclease to virtually any sequence in the human genome. Once there, Cas9 cuts the DNA double helix and the cell's own repair machinery takes over. If left alone, the break is sealed imperfectly, introducing small insertions or deletions that can disable a gene. But if provided with a carefully crafted DNA template, the cell may repair the cut by copying the new sequence into place. Thus, in principle, any gene can be disrupted, corrected, or even rewritten. The implications are staggering. Monogenic diseases, those cruel conditions born of a single mutated gene, suddenly appear vulnerable. Disorders like sickle cell anemia, cystic fibrosis, or Duchenne muscular dystrophy, once deemed incurable, now shimmer on the horizon of possibility. Already, clinical trials have harnessed CRISPR to edit blood stem cells ex vivo, correcting defective genes before reintroducing them into patients. For the first time, cures seem not a distant fantasy but a tangible pursuit. Yet, the power of gene editing extends far beyond therapy. Scientists wield it to engineer animal models of disease, to unravel gene function with unprecedented speed, to design crops resilient against climate and blight, and even to contemplate synthetic genomes. With CRISPR and its variants Cas12, Cas13, base editors, prime editors, the genome is no longer a static text but a manuscript open to revision. But such power is never without peril. Editing a gene in a somatic cell to cure an individual is one matter. Editing the germline so that alterations are passed to future generations is quite another. Here, the ethical dilemmas grow vast and shadowed. What if gene editing, instead of curing disease, were used for enhancement? What if the human genome became a canvas for vanity, inequality, or hubris? These questions stir fierce debate, reminding us that while the technology is precise, human intent may not always be so noble. Moreover, even in the laboratory, challenges remain. Off-target effects, cuts made at unintended sites still haunt the technology, threatening unforeseen consequences. Mosaicism, in which not every cell receives the desired edit, complicates outcomes. And while delivery into cells ex vivo can be controlled, delivering CRISPR machinery safely and efficiently into tissues within the body remains a formidable obstacle. Yet, despite these caveats, the trajectory of gene editing is unmistakable. It is advancing with a velocity rarely seen in biomedical science, and each year yields refinements, new enzymes, and greater precision. The dream of rewriting life's text, once the province of poets and prophets, is now the task of biologists. In the context of pathology, gene editing is more than a technical marvel, it is a conceptual revolution. No longer are we confined to describing the errors of nature, we now possess the tools to repair them. Disease may soon be addressed not at the level of symptoms, or even proteins, but at the source code itself. To alter a single nucleotide may mean to rescue a life, to silence a devastating inheritance, to free future generations from ancestral burdens. As we stand on this threshold, the genome appears less like a fate inscribed in stone and more like a manuscript in clay, malleable, revisable, and perhaps, one day, entirely designable. Whether this will usher in an age of healing or hubris remains to be seen, but one truth is undeniable: the pen is now in human hands. From this grand ability to write and rewrite, we must return to the ordinary yet essential tasks that allow cells to survive in the first place, the mundane housekeeping chores without which life would crumble. For before genomes can be edited, messages regulated, or signals transmitted, a cell must feed, clean, and sustain itself. And it is to these labors of cellular housekeeping that we now turn. Cellular housekeeping. Though the cell may dazzle us with its feats of signaling, its orchestras of transcription, and its revolutions of division, it is not sustained by majesty alone. Beneath the fireworks of biology lies an economy of chores, routines as vital as they are unglamorous. Just as a city cannot flourish without sanitation, maintenance, and delivery of food and water, so too does the cell require order in its internal housekeeping. Without these everyday labors, the grand symphonies of DNA and proteins would collapse into cacophony. Consider first the intracellular trafficking of proteins, for proteins are not simply manufactured, but must be shepherded to their destinations. Enzymes destined for the lysosome, receptors bound for the plasma membrane, transcription factors returning to the nucleus, each must be guided by signal sequences, tags written into their amino acid chains like addresses upon letters. Without these signals, proteins would wander aimlessly, clogging the cytoplasm, colliding into structures where they do not belong. Then there is the indispensable task of degradation. A cell that only accumulates and never discards would suffocate under its own refuse. Thus exist mechanisms of selective destruction, each ensuring that worn-out or misfolded proteins are dismantled and recycled. The ubiquitin proteasome system tags such proteins with chains of ubiquitin, marking them for delivery into the proteasome's hollow cylinder, where they are unfolded, chopped, and rendered back into amino acids. Parallel to this is the system of autophagy, whereby whole organelles, protein aggregates, or invasive microbes are engulfed within double-membraned vesicles that fuse with lysosomes, allowing hydrolytic enzymes to perform their work. Autophagy is no mere disposal, it is also survival, a way for cells under starvation to cannibalize their own stores and sustain themselves until nourishment returns. Equally vital is the maintenance of ionic gradients and osmotic balance, achieved by pumps and exchangers embedded in the plasma membrane. Sodium, potassium, calcium, hydrogen all are regulated with meticulous care. This regulation is not merely structural, but functional. Muscle contraction, nerve transmission, secretion all hinge upon such ionic equilibria. Lipid metabolism, too, forms a cornerstone of housekeeping. Within the smooth endoplasmic reticulum, lipids are synthesized, modified, and distributed. These molecules furnish membranes, fuel energy storage, and create signaling mediators. In the absence of this lipid economy, membranes would falter, organelles would crumble, and signaling pathways would stutter into silence. One must also not forget the careful regulation of oxidative stress, for metabolism inevitably produces reactive oxygen species, byproducts as corrosive as sparks in a library. Cells combat this with antioxidants, glutathione foremost among them, and with enzymes such as superoxide dismutase and catalase. When balance is lost, when reactive species outpace defenses, cellular components oxidize, membranes are breached, and DNA itself is scarred. Here, too, the ordinary labor of scavenger radicals becomes the very shield against degenerative disease. Thus, the cell is more than its nucleus, more than its glamorous mitochondria or intricate signaling cascades. It is a home, ceaselessly tended by systems of maintenance, recycling, and repair. Without these, the edifice of life would not merely weaken but collapse. Having surveyed these domestic labors, it is fitting to turn next to the great gatekeeper of the cell, the plasma membrane. This delicate yet formidable barrier is both protector and mediator, holding the outside world at bay even as it welcomes the nutrients and signals upon which survival depends. Plasma membrane, protection and nutrient acquisition. If the genome is the script and the cytoplasm its stage, then the plasma membrane is the curtain that both reveals and conceals. It is at once guardian and gate, custodian of integrity and mediator of exchange. Without this thin, shimmering veil of lipids and proteins, the cell would dissolve into the ocean of the extracellular world, losing its identity, its coherence, its very existence. With it, the cell is sovereign, distinct, and alive. The plasma membrane is a paradox of elegance and strength. Barely 7 to 10 nanometers in thickness, it is constructed upon the principle of the lipid bilayer, a double sheet of amphipathic molecules whose hydrophobic tails face inward while hydrophilic heads greet the aqueous surroundings. This bilayer, fluid and dynamic, provides the structural foundation. Yet the membrane is no inert partition. Embedded within it are proteins of astonishing variety, channels, carriers, receptors, enzymes, all of which transform the boundary into a bustling marketplace of exchange. In its protective role, the plasma membrane functions as the sentinel against chaos. It shields the cytoplasm from the indiscriminate influx of ions and toxins, ensures that metabolic intermediates remain corralled, and provides mechanical resilience against osmotic stress. At the same time, it is also exquisitely selective in what it allows across. Life is not sustained by isolation, but by controlled commerce, and it is here that the membrane assumes its second, equally vital role: nutrient acquisition. Nutrients, growth factors, hormones all approach the membrane like supplicants at a gate. Some, small and lipophilic, slip easily through the lipid bilayer by diffusion. Others, charged or bulky, must be ferried by proteins each specialized for its cargo. Glucose, amino acids, nucleosides all enter by way of specific transporters. Lipoproteins, with their cargo of cholesterol and triglycerides, are welcomed through receptor-mediated endocytosis. Even whole pathogens exploit these portals, masquerading as nutrients to gain illicit entry. Endocytosis and exocytosis extend the membrane's repertoire, allowing it to engulf or expel cargo in vesicles. Through endocytosis, the cell internalizes ligands, receptors, and nutrients. Through exocytosis, it dispatches neurotransmitters, hormones, and enzymes. In this ceaseless traffic, the membrane is less a barrier than a bustling harbor, with ships docking and departing, unloading sustenance and exporting signals. Yet the membrane's role is not limited to transport alone. It is also the arena of recognition and communication. Glycoproteins and glycolipids studding its surface create a molecular signature, the glycocalyx, by which cells recognize one another, distinguish self from non-self, and interact with their neighbors. Immune cells probe these signatures relentlessly, deciding whether to grant tolerance or to unleash attack. Pathogens, too, exploit them, binding to specific surface molecules as their entry points. In the grand design of the cell, therefore, the plasma membrane is no mere wall, but a living interface, flexible, dynamic, and richly adorned with proteins. It is the place where identity is declared, nutrients are secured, and signals from the outside world are first received and interpreted. From this boundary, we must move inward into the processes that allow the membrane to fulfill its role as gatekeeper, the sophisticated mechanisms of membrane transport, by which ions, molecules, and even macromolecules are shuttled with astonishing specificity and precision. Membrane transport. If the plasma membrane is the wall of the city, then its transport systems are the gates and aqueducts that sustain life within. No fortress, however mighty, can survive in absolute isolation. It must control what enters and what leaves. So too with the cell. To live, it must import nutrients, expel wastes, and regulate ions with precision so exacting that even slight deviations may spell disaster. The bilayer itself is inherently selective. Small, nonpolar molecules such as oxygen and carbon dioxide slip across effortlessly, guided only by concentration gradients. Water, too, though polar, finds passage through specialized channels known as aquaporins, allowing it to rush where osmotic currents dictate. But larger or charged molecules cannot simply drift through this lipid sea. They require the assistance of proteins embedded in the membrane, proteins that serve as ferries, tunnels, or pumps depending on circumstance. The simplest form of transport is passive diffusion, driven solely by gradients. Here, the cell expends no energy, for molecules flow from regions of abundance to scarcity, seeking equilibrium. Glucose transport into many cells, for instance, is mediated by carrier proteins that facilitate this passive passage. Yet, equilibrium is not always sufficient. Life demands asymmetry, gradients that fuel processes and encode information. For this, the cell harnesses active transport mechanisms that consume energy, often in the form of ATP, to drive molecules against their gradients. The sodium-potassium pump is the exemplar of this labor. With each cycle, it expels 3 sodium ions from the cell and brings in 2 potassium ions, consuming ATP in the process. The result is a steep gradient of sodium outside and potassium inside, a disequilibrium essential for nerve impulses, muscle contraction, and nutrient transport. Other pumps, such as calcium EPases, maintain the low cytoplasmic calcium concentration required for signaling. When calcium surges inward through channels, it acts as a second messenger, triggering contraction, secretion, or gene transcription. But after the surge, pumps labor tirelessly to restore the resting state, ensuring that the signal is sharp and transient rather than dull and persistent. The membrane also employs secondary active transport, a clever exploitation of existing gradients. Here, the downhill flow of one molecule powers the uphill movement of another. The sodium-glucose cotransporter in the intestinal epithelium is a striking example. Sodium rushing in along its gradient drags glucose with it, enabling absorption of life's simplest fuel even against concentration difference. Beyond small molecules and ions, the cell must also import and export larger cargo. This is the domain of vesicular transport, endocytosis and exocytosis. Endocytosis allows the plasma membrane to invaginate, engulfing extracellular fluid, receptors, and ligands into vesicles. In receptor-mediated endocytosis, a ligand binds to its receptor, clusters into clathrin-coated pits, and is internalized with exquisite specificity. Cholesterol uptake, mediated by LDL receptors, follows this path. When this system fails, as in familial hypercholesterolemia, cholesterol accumulates in blood, reminding us of how vital transport is to homeostasis. Exocytosis, in contrast, dispatches cargo outward. Secretory vesicles fuse with the plasma membrane, releasing neurotransmitters at synapses or insulin from pancreatic beta cells. This mechanism is so finely tuned that in neurons, vesicles dock, prime, and then await the whisper of a calcium influx before fusing, ensuring communication is swift and precise. It is through this array of transport systems, diffusion, carriers, pumps, vesicles, that the cell conducts its commerce. The balance is delicate. Too much or too little flux spells dysfunction. Cystic fibrosis, for instance, arises from defective chloride channels, leading to viscous secretions that compromise lungs and pancreas. Such diseases are stark reminders that transport is not a peripheral detail, but a central pillar of cellular life. From the membrane and its gates, our exploration must now extend inward to the cytoskeleton, the invisible scaffolding that not only shapes the cell, but also anchors these very transport processes, guiding vesicles like cargo along railways to their destinations. Cytoskeleton. The cytoplasm is no chaotic soup, no formless slurry where organelles drift at random. It is an ordered space, sustained by a web of filaments that lend both architecture and dynamism. This is the cytoskeleton, a lattice of protein polymers that holds the cell in shape, anchors its contents, and enables motion both within and without. To call it a skeleton is to understate its vitality, for unlike the rigid bones of an organism, this framework is alive with ceaseless remodeling, ever adapting to the needs of the cell. The cytoskeleton is woven of three great classes of fibers: actin filaments, microtubules, and intermediate filaments, each with its own character, its own role in the orchestration of cellular life. Actin filaments, also called microfilaments, are the most delicate yet the most versatile. Composed of thin strands of actin protein, they form dense networks beneath the plasma membrane, lending it resilience and shape. It is actin that drives the crawling of cells across a surface, pushing forth lamellipodia and filopodia like exploratory fingers. It is actin that pinches the cell in two during cytokinesis, tightening a contractile ring until the daughter cells separate. And it is actin that links membrane proteins to the cytoskeleton, transmitting signals from the surface inward and stabilizing contacts with neighboring cells. Though slender, these filaments are powerful, for they can polymerize and depolymerize within seconds, reshaping the cell with fluid agility. If actin is the artisan, then microtubules are the highways. Built from tubulin dimers, these hollow cylinders radiate outward from the centrosome like spokes from a hub, forming polarized tracks along which motor proteins ferry cargo. Dynein and chysin, the beasts of burden, stride along these rails, carrying vesicles, mitochondria, even entire chromosomes during mitosis. Without microtubules, the cell would be paralyzed, its organelles stranded, its chromosomes unable to segregate. The most dramatic manifestation of microtubule architecture is the cilium and the flagellum whip-like appendages, whose coordinated beating propels cells, or moves fluid across their surfaces. Within their cores, the 9+2 arrangement of microtubules bends rhythmically under the force of dynein, a belay that sweeps mucus in the airway or drives the spermatozoon toward its destiny. Intermediate filaments, finally, are the stalwart girders of the cytoskeleton. Unlike actin or tubulin, they are not dynamic rails of transport, but resilient cables that bear tension and lend mechanical strength. Composed of proteins that vary with tissue: keratin in epithelia, vimentin in mesenchyme, desmin in muscle, neurofilaments in neurons, they provide identity as well as endurance. When the cell is stretched, it is intermediate filaments that resist rupture. When tissues are stressed, they link cells together at desmosomes, uniting them into cohesive sheets. The clinical consequences of their failure are grave. In epidermolysis bullosa, mutations in keratin render the skin fragile so that the lightest touch produces blisters. Together, these three systems create not only a framework, but also a choreography. Actin reshapes membranes, microtubules direct long-range traffic, intermediate filaments reinforce stability. They are constantly cross-talking, coordinated by signaling pathways that sense the environment and remodel the cytoskeleton in response. The result is a cell that is not rigid but supple, not static but alive with possibility. The cytoskeleton is more than a scaffold, it is a force of motion. It enables cells to migrate in development and healing, to engulf microbes in phagocytosis, to divide, to sense their neighbors. In cancer, cytoskeletal dynamics are hijacked, allowing malignant cells to invade and metastasize. In neurodegeneration, the breakdown of microtubules and their associated proteins cripples axonal transport, starving neurons of sustenance. Thus, the cytoskeleton is not an accessory to life, but one of its most central foundations. From the filaments that shape and move the cell, we must next pass to the junctions that bind cells together and allow them to speak with one another. For no cell is an island, each lives as part of a community, held by cell-cell interactions that integrate tissues into organs and organisms. Cell-cell interactions. A single cell is remarkable, but an organism is miraculous. What transforms countless individual cells into the harmony of tissue and organ is not merely their structure, nor merely their function, but their ability to connect, to adhere, and to communicate. Cell-cell interactions are the mortar of multicellular existence, fastening cells into coherent assemblies while simultaneously allowing the whispers of communication to flow across their boundaries. The simplest requirement of tissues is cohesion. Cells must cling to one another and to the extracellular matrix lest they drift apart. This cohesion is achieved by cell junctions, protein complexes that rivet membranes together with architectural precision. Among them, tight junctions form the seals. Found near the apical surface of epithelial cells, they fuse membranes into barriers impermeable to solutes, ensuring, for example, that gastric acid does not seep between the lining cells of the stomach. Adherens junctions and desmosomes, in contrast, are anchors. They link the cytoskeletons of adjacent cells. Actin in adherens junctions, intermediate filaments in desmosomes, thereby distributing mechanical stress across the tissue. Without them, epithelia would tear apart at the lightest strain. Finally, gap junctions provide not adhesion but communication, tiny channels that allow ions and small molecules to pass directly from one cell to another. Through these pores, cardiac muscle cells synchronize their contractions, neurons coordinate glial partners, and developing tissues share gradients of signaling molecules. The proteins that underlie these junctions are as varied as the junctions themselves. Caterins, calcium-dependent adhesion molecules, mediate adherens junctions and desmosomes. Claudins and occludins seal tight junctions. Connexins form the hexameric channels of gap junctions. Each belongs to a family of proteins evolved to transform membranes from mere boundaries into points of union. But adhesion is not mechanical alone, it is also informational. The moment two cells touch, their junctions become platforms of signaling. Caterins, integrins, and other adhesion molecules transmit information inward, altering cytoskeletal dynamics, gene expression, even cell survival. The decision of a stem cell to proliferate or differentiate, of an immune cell to attack or retreat, often depends on the signals it receives at points of contact. Thus, adhesion is not passive but conversational, encoding instructions about position, identity, and destiny. The importance of these interactions is revealed most vividly when they fail. Loss of adhesion is one of the hallmarks of cancer progression. Malignant cells downregulate E-caterin, loosen their bonds to neighbors, and slip free of epithelial sheets. Once detached, they invade surrounding stroma, enter circulation, and colonize distant organs. Diseases such as pemphigus vulgaris, in which autoantibodies attack desmosomes, demonstrate another dimension. Tissues literally fall apart, blistering with fragility when adhesion molecules are compromised. Equally striking is the role of cell-cell communication via gap junctions and surface receptors. In the heart, ions passing through connexin channels ensure rhythmic synchrony of contraction. When these connexins fail, arrhythmias ensue. In the brain, glial networks connected by gap junctions buffer potassium and distribute metabolites. Even in the immune system, transient interactions between lymphocytes and antigen-presenting cells formed by integrins and co-receptors direct the very fate of immune responses. Cell-cell interactions also mediate the architecture of development. During embryogenesis, gradients of adhesion molecules guide the sorting of cells into layers and tissues. Cells, expressing similar caterins cluster, segregating into distinct populations. Without this selective adhesion, tissues would remain formless and the exquisite choreography of development would collapse. Thus, cell-cell interactions are more than mere glue. They are the language by which cells recognize kin, the locks that ensure tissues are sealed, and the portals through which signals are exchanged. They transform individuality into community, solitude into society. From the adhesion and dialogue between neighbors, we must now turn to the signals that travel not only between touching cells, but also across distances, the grand realm of cell signaling, in which molecules act as messengers to coordinate physiology across the body. Cell signaling. If adhesion is the glue of multicellularity, signaling is its voice. No cell, however intricately structured, can exist in splendid isolation. Survival, differentiation, proliferation, and death all are dictated by signals that cells both emit and interpret. The orchestration of life depends upon this biochemical symphony in which molecules serve as messengers and receptors act as listeners, ensuring that the trillions of cells within an organism function not as anarchic individuals, but as members of a coherent whole. At its core, cell signaling is an information network composed of three essential stages. A signal must be generated, received, and transduced into a cellular response. The signal may be a soluble molecule, a membrane-bound ligand, or even a mechanical stimulus. The receiver is almost always a receptor protein exquisitely attuned to detect it. Upon binding, the receptor does not merely absorb the message, it translates it, launching cascades of intracellular events that ripple through cytoplasm and nucleus alike. The modes of signaling are diverse, each suited to the context of communication. In autocrine signaling, a cell speaks to itself, releasing signals that bind its own receptors, in intimate feedback loop crucial in immune cells and tumor biology. Paracrine signaling extends communication to immediate neighbors, guiding tissue development and local responses. Neurotransmitters crossing the synaptic cleft are its most dramatic example. At greater distances, endocrine signaling dispatches hormones into the bloodstream, allowing endocrine glands to whisper commands that coordinate physiology across the entire body. A more tactile form, juxtacrine signaling, requires direct contact between cells, often mediated by membrane-bound ligands, particularly vital during embryonic patterning. At the molecular level, receptors stand as the sentinels of signaling. Some, like G-protein-coupled receptors GPCRS, reside at the membrane surface, recognizing extracellular messengers and triggering cascades through second messengers such as cAMP, IP, and calcium ions. Others, such as receptor tyrosine kinases RTKS, convert ligand binding into phosphorylation events, propagating signals via MAP kinase pathways that influence growth and survival. Still others, like nuclear receptors, lie dormant within the cytoplasm or nucleus, activated only when lipid-soluble signals such as steroid hormones diffuse across membranes to bind them. Each receptor type encodes its own dialect of cellular instruction, ensuring specificity amid the cacophony of circulating signals. Once engaged, signals are amplified through cascades, molecular relay races in which one activated protein triggers the next, exponentially magnifying the original cue. This architecture provides not only sensitivity but also points of regulation. Crosstalk between pathways allows integration of multiple inputs, ensuring that no single signal dictates fate in isolation. Thus, a cell's decision to divide, differentiate, or die emerges as the weighted sum of numerous intersecting pathways. The consequences of signaling are vast. Growth factors drive the proliferation of epithelial sheets, chemokines summon leukocytes to sites of infection, morphogens sculpt the form of developing embryos, and apoptotic signals ensure that defective or surplus cells bow out gracefully for the greater harmony of the organism. Yet, when signaling falters, the results are catastrophic. Mutations that lock receptors in odd positions unleash unrestrained growth, fueling cancers. Defects in insulin signaling lie at the heart of diabetes. Aberrant neurotransmitter signaling underpins psychiatric and neurodegenerative disorders. Beyond pathology, signaling is the key to adaptability. Cells continuously recalibrate their sensitivity by upregulating or downregulating receptors, desensitizing after prolonged exposure, or sensitizing to rare stimuli. Such plasticity ensures that cells remain responsive, not overwhelmed by their environment. In this sense, signaling is not static instruction but a dynamic conversation, modulated by context, time, and history. Indeed, one might say that signaling confers upon cells a rudimentary form of perception. They sense hormones as an animal senses light or sound. They interpret gradients as a traveler interprets landmarks. They act upon information, sometimes in milliseconds, sometimes over hours or days. Signaling bestows on cells a temporal awareness, enabling them to anticipate, adapt, and survive. Thus, cell signaling is more than molecular mechanics. It is the governance of life itself, the dialogue that makes multicellular existence intelligible. From the solitary cry of autocrine feedback to the systemic decree of endocrine hormones, signals weave individual cells into the living fabric of organisms. Maintaining cell populations, proliferation and the cell cycle, the perpetuation of tissues, the renewal of worn cells, and the healing of wounds all depend on a single principle: cell proliferation. Yet, this process is no blind multiplication. It is an exquisitely ordered sequence known as the cell cycle, designed to ensure that the genetic patrimony is copied with precision and distributed with fidelity. The cycle unfolds in phases, each a gate that must be properly passed before the next may open. In G1 phase, the cell grows, sensing its environment, gauging nutrient supply, and receiving extracellular signals. Here lies the restriction point, a critical decision node where the cell commits either to enter S phase or to withdraw into quiescence G. S phase is the domain of DNA synthesis, where each chromosome is faithfully duplicated. G2 phase follows, a period of proofreading and preparation in which the machinery checks the replicated genome for errors before embarking upon division. At last comes M phase, mitosis, where the sister chromatids are separated and apportioned into daughter cells. Cytokinesis completes the process, yielding two progeny poised to begin their own cycles. This orderly sequence is safeguarded by checkpoints, molecular sentinels, that ensure fidelity. The G/S checkpoint verifies that DNA is intact and conditions favorable. The G/M checkpoint ensures replication is complete. The spindle checkpoint monitors attachment of chromosomes to the mitotic spindle. At the heart of these controls are cyclins and their partners, the cyclin-dependent kinases CDKs. Cyclins rise and fall with the phases of the cycle, activating CDKs, which phosphorylate key proteins to drive progression. The tumor suppressor 53, guardian of the genome, halts the cycle in the presence of damage, buying time for repairor if damage is irreparable, steering the cell toward apoptosis. The balance of proliferation is vital. Too little division results in atrophy and impaired healing, too much in hyperplasia and neoplasia. Cancer, at its essence, is disease of the cell cycle. Mutations in oncogenes that accelerate division, or in tumor suppressors that normally restrain it, yield unchecked growth. Conversely, targeted manipulation of the cell cycle offers therapeutic promise, as seen with CDK inhibitors in cancer therapy. Thus, the cell cycle is both the engine of renewal and a point of vulnerability, the rhythm by which populations of cells are maintained in health and distorted in disease. Stem cells. At the heart of tissue maintenance and renewal lies a most remarkable class of cells, the stem cells. Unlike their differentiated progeny, which are committed to specific tasks, stem cells embody potential. They possess the dual gifts of self-renewal, the ability to divide and replenish themselves indefinitely, and potency, the capacity to give rise to more specialized cell types. Together, these properties endow them with a central role in sustaining the cellular populations of the body across a lifetime. Stem cells are classified according to their range of developmental potential. Totipotent stem cells, exemplified by the zygote and its earliest descendants, can generate every cell type of the body as well as the extraembryonic tissues. Pluripotent stem cells, such as those of the inner cell mass of the blastocyst, can form any tissue of the embryo proper but not the placenta. More restricted still are multipotent stem cells, capable of producing only the lineages within a particular tissue or organ. Adult tissues harbor these multipotent progenitors, hematopoietic stem cells in the bone marrow that continuously generate blood cells, or mesenchymal stem cells that yield bone, cartilage, and adipose tissue. Finally, unipotent stem cells possess the narrowest fate, giving rise to a single cell type, but retaining the essential feature of self-renewal. Within tissues, stem cells are often sequestered in specialized microenvironments termed niches. These niches provide the cue signals from neighboring cells, contact with extracellular matrix, gradients of growth factors that preserve stemness or trigger differentiation as needed. The crypts of the intestinal epithelium, where LGR5+ stem cells fuel rapid epithelial turnover, and the bulge region of the hair follicle, where quiescent stem cells awaken during hair cycling, exemplify the exquisite regulation provided by such niches. The existence of stem cells also reconciles two seemingly opposite needs, stability and plasticity. They maintain stable cell populations in tissues such as skin and blood, where cells are constantly shed and replaced. Yet, they also furnish plasticity in response to injury, awakening dormant reserves to regenerate damaged structures. The liver's regenerative prowess, for instance, depends not only on the proliferation of surviving hepatocytes but also on facultative stem-like cells that can be mobilized when conventional repair is insufficient. In pathology, the stem cell paradigm carries profound implications. Cancer stem cells, a subpopulation within tumors, are thought to sustain malignancy, resist therapy, and seed relapse. Conversely, depletion or senescence of normal stem cells contributes to aging as tissues lose their capacity to renew. Understanding the biology of stem cells, how self-renewal is balanced against differentiation, how potency is constrained or released, thus lies at the core of both regenerative promise and oncologic peril. Regenerative medicine. From the recognition of stem cell potential has emerged one of the most ambitious enterprises of modern science, regenerative medicine. Its goal is nothing less than to restore, replace, or regenerate damaged tissues and organs, moving beyond palliation toward true repair. At its foundation is the harnessing of stem cells. Embryonic stem cells, ESCs, with their pluripotency, offer the ability to generate virtually any cell type. Yet, their use is fraught with ethical considerations and the risk of teratoma formation. A transformative advance came with the discovery of induced pluripotent stem cells, iPSCs, created by reprogramming adult somatic cells back into a pluripotent state. iPSCs bypass many ethical barriers and open the possibility of patient-specific therapies in which one's own cells could be re-engineered to replace damaged tissues without risk of immune rejection. Applications of regenerative medicine are already unfolding. In hematology, bone marrow transplantation, a therapy grounded in the principles of stem cell biology, has long been used to cure leukemias and congenital immunodeficiencies. Advances in culture techniques now allow the generation of cardiomyocytes from pluripotent stem cells, raising the prospect of repairing damaged myocardium after infarction. In ophthalmology, trials are underway using stem cell-derived retinal pigment epithelium to treat macular degeneration. Even complex tissues such as skin, cartilage, and trachea have been bioengineered and transplanted, proof that the union of stem cell science with biomaterials can yield functional replacements. Regenerative medicine also looks beyond cell replacement. Gene editing technologies, such as CRISPR-Cas9, when combined with stem cells, permit the correction of genetic defects at their root, offering cures rather than symptomatic treatments. Likewise, advances in tissue engineering aim to recreate not only cell populations but the structural and biochemical milieu, scaffolds, extracellular matrix, vascular networks necessary for long-term integration and function. Challenges remain formidable. Controlling differentiation with precision, ensuring safety from mutations or tumorigenesis, and scaling laboratory success to clinical reality all demand careful navigation. Immune rejection, even with autologous cells, can occur if subtle genetic or epigenetic changes arise. Ethical and regulatory frameworks must balance innovation with protection. Yet, despite these barriers, the trajectory of progress is unmistakable. At its deepest level, regenerative medicine is more than therapy, it is a philosophical shift. Where once degeneration and loss were accepted as irreversible, biology now offers a vision of renewal. The ability to reawaken stemness, to reshape fate, to rebuild what was broken, gestures toward a future in which the boundary between healing and creation is blurred. Conclusion of maintaining cell populations. Thus, in the maintenance of cell populations, we glimpse the cycle of biological continuity. Through the disciplined rhythm of the cell cycle, populations are renewed. Through the latent potential of stem cells, tissues are preserved and repaired, and through the daring enterprise of regenerative medicine, humanity aspires to harness these principles for restoration beyond the limits of natural healing. In these processes, the fundamental truth of biology is reaffirmed, that the life of organisms is sustained not by permanence, but by continuous renewal.